We're moving on now to the presentation from Mitochon Pharmaceuticals, and the speaker will be John Giesler, who will be talking about microdoses of mitochondrial uncoupler MP101 in hereditary SOD1 G93A, providing the first ever small molecule with functional recovery from paralysis and hints that ALS is a solvable problem. So,
great.
Thanks for John.
James, thank you. Jen, thank you for setting this up. Great opportunity. So 2024 is a big year for us. We're in the clinic with ALS patients, and we have this really provocative data that on the SOD1 mouse that shows a reversal of paralysis with this mitochondrial small molecule, which is, I'm told, the first ever in the SOD1 mouse model. So without any delays. So in 8th grade biology, you were told the mitochondria is the powerhouse of the cell. What they didn't get into is the mitochondria is also involved in energy calcium homeostasis. It is primary job is to keep the cytosol clear of calcium and to store calcium at huge levels. The other thing is that the mitochondria, with its due to the electron transport chain pumping out protons, there's 180 millivolts difference between the outside and the inside. The inside is pH basic, and it's the only organelle out of all the organelles with a pH basic environment. And that's important because our drug's a weak acid and specifically drawn to the mitochondria. And the other thing is that the mitochondria are extremely evolutionarily conserved among mammals. So we work with a drug that was used 100 years ago called 2,4-dinitrophenol. This is our platform. It was used 100 years ago before the FDA approved it for anything. It was never in a GLP tox study 100 years ago. They didn't even know how the mitochondria worked or how the drug worked, but it was used for obesity back in the 1930s. Hundreds of thousands of people used it. It was quite effective for weight loss, but it also had issues at high doses. And that has nothing to do with what we're doing today. Our doses are all micro doses, weight neutral, weight preserving. And then the thing that I want to take home today is that this is a completely different program than has ever been seen for ALS patients or neurodegenerative diseases. This is based on a proton therapy. You're used to ASOs and other small molecules. This is completely different. So MP101 is a weak acid. It has a dissociable proton due to the pulling away of the nitro groups of that electron off the hydroxyl group. So the hydrogen there, which is a pure proton, is weakly held on, goes to the only organelle out of all the organelles with the pH basic environment, directly and specifically targets the mitochondria, releases that proton into the mitochondrial matrix, changing the membrane potential and the physiology of that mitochondria. The pharmacology is considered non-genomic, meaning that the target is a location. It's the mitochondrial matrix itself. It doesn't target the enzymes or the RNA or DNA. Specifically is modulating the mitochondrial membrane potential. And it prevents this reverse flow of electron in the electron transport chain, which is the source of overt free radicals. And the other thing it does, which is voltage gated, is it closes the uniporter, preventing calcium influx, calcium overload, and which is, if it's not under control, it's the kiss of death for the cell. And then also MP101 changes the physiology of about 2,000 mitochondria per the population of a cell, moving it towards a better population as an oral once per day, brain penetrating small molecule that is at microdoses is pro-survival. So there's three things I want you to take home today on the pharmacology is that one, that the mitochondrial matrix is extremely sensitive to free radical productions under the presence of under an uncoupler. Two is that the mitochondria under a DNP or free uncouplers closes the uniporter. It increases the cytosolic calcium, but it lowers mitochondrial matrix calcium. And this event lowering the mitochondrial calcium improves cellular stability and homeostasis of the calcium, and that prevents the triggering of apoptosis. MP101 also in DNP lowers mTOR, changing the autophagy, mitophagy. And the third part I want to say is that there's an induction of BDNF, brain-derived neurotrophic factor, in the cortex. This is a mouse model, wild type, 2 weeks of dosing once per day, and taking the cortex out and measuring how much BDNF is in the cortex or the striatum, cerebellum. And there's a nice dose response that goes up. And for BDNF in the brain, it's considered the fertilizer of the brain, involved in longevity and cognition and cognitive properties. Below the neck, it's actually a myokine involved in islet protection, muscle nourishment, and protection. So those are the three things. So if you take this little cartoon that I have here to talk about the pleiotropic effects of mitochondrial uncoupling. So MP101 is DNP, MP201 is a prodrug of DNP. If MP101 goes into the mitochondrial matrix, it releases the hydrogen and that it's a pure proton. It comes out, the MP101 as an anion, gets reprotonated back in the acidic environment of the cytosol, goes back in and drops off another proton. Eventually, it's eliminated by in urine as a whole molecule. This event lowers free radicals, changes calcium flux into the mitochondria. And this event, which purely is based on biophysics, cascades into activation of adenylate cyclase, which generates cyclic AMP. The transcription for CREB is activated, and CREB is the transcription factor for BDNF. So you have this lowering of damage with a promotion of repair that actually moves into genomic remodeling of the cell. So several years ago, this very new scientist, she's a postdoc at Rochester at the time, Dr. Lanwi Lepere, she had this central hypothesis, and that she calls, and it's the idea that she believed that it's both the toxicity in the skeletal muscle and in the neuron that plays a primary role in causing the nerve to lift off and cascading into ALS. So she approached us to tell us that our mitochondrial couplers could have a profound effect on neuroprotection and neuromuscular protection by the fact that it lowers calcium influx. So she took on this idea, and we provided her the drug. This was the data that she first got. This is an early onset of in the SOD1 mouse. This is 0.5 MPK, which is about 2 mg per day in a human. Starting at 4 weeks old and going out to 15, she could show that the purple mouse, which is the MP101 treated SOD1, is basically equivalent to the black, which is the wild type, in the ability to stay on the rotor rod for 60 seconds with varying RPMs. So we're somewhere around 30 RPMs. The vehicle-treated SOD1 fall off, but out to 37 or beyond, MP101 treated mice stay on quite well. And then recently, she generated some nice data to show that the neuromuscular juncture is and muscle innervation is intact. So now these are 10 weeks old animals that were treated till 18 weeks. So 18 weeks is shown here to show that there's a nice preservation of the neuromuscular juncture in the shown in purple compared to the vehicle in orange. Very and quite significant for almost as approaching the control. And then in muscle, in EDL, she shows that there's a reduction in inflammation going from orange to blue for MP101, changes in protein carbonyl with oxidation of proteins, and there seems to be an induction of muscle synthesis with MP101. So there's a nice effect on what we believe the pharmacology of MP101 actually does. So this is the data that's really quite striking, and this is the basis of the idea that, so this is that the first molecule to ever have functional recovery and paralysis. So here is the SOD1 mice. She started treating them at 10 weeks old. On the top is the vehicle-treated wild type, and the bottom are all the SOD1 mice. And at 0.5 MPK group, there is a point in which the animals in green are running, and then in red not running. But then 6 out of the 10 animals began running again for either 1 to 3 weeks, and then they stopped running again. There was one mouse that never failed running. There's 3 mice that once they stopped running, never ran again. If you compare that to the vehicle-treated SOD1, all the mice, once they stopped running, they never ran again. So if you put this into a Fisher's exact test, the p-value is 0.01, which is categorical data that suggests that this is quite significant in the ability to recover from paralysis. So the paper was just accepted like 2 weeks ago. We're pretty excited, and this should be out in probably about a month, showing this the data and the functional recovery in the SOD1 mouse. So before I get into the clinical part of the program, I need to introduce you to our favorite biomarker, which is isoprostanes. And isoprostanes hopefully will become your favorite biomarker. So this is a small molecule, this is a lipid, arachidonic acid. And when free radicals are generated, they attack DNA, they attack proteins, but when they attack lipids, and particularly this particular population of lipids, it puts a double bond in the lipid, and isoprostanes are then generated. They are very, very small lipid. They come out in your urine, your blood, your fluids, your cerebrospinal fluid, your tears, your spit, your bronchial fluids, and so this is our biomarker for an indirect readout that mitochondrial free radicals have changed. And it has a 16-minute half-life in humans. So when we have our clinical studies, we have plenty of time to flatten isoprostanes. So I wanted to show you a video in a different movement disorder. This is the MS model which shows the mouse in the peak of paralysis is completely paralyzed. This is 7 doses of treatment of MP101 before the peak of paralysis. And then here's the prodrug, 16 MPK, and then the higher dose of the prodrug of MP101, DMP. And we go from a mouse that is completely paralyzed to the bottom of the cage to one that is able to hop out of the cage, which is, I believe, quite striking. And then the reason to show you this is also to show you that what happened to the isoprostane. So the mouse that you saw paralyzed at the bottom of the cage has isoprostane levels in the spinal column of 316. 7 doses of MP101 or MP201 completely flattened isoprostanes to a p-value of 10 to the -6, almost equivalent to the wild type non-treated control. So this is our biomarker going into the clinic. So the clinical study that we have planned is a basket study. They have a 14-day study that's been approved in Europe. Take the human participants with ALS, MS, Huntington's, and Alzheimer's, and we will take the cerebrospinal fluid at day 0. Then there's 14 days of oral capsules of MP101 once per day, and then we'll take the cerebrospinal fluid again at day 15 to look at safety, tolerability, and then a whole portfolio of biomarkers. So the basic idea is we take the patients are screened by their neurologist for their inclusion-exclusion criteria. If they pass that, then their urine goes off to a lab in Berlin to look at, by LC mass spec, the amounts of isoprostanes in the urine. And there's a 95% confidence that if you're above 3.75, then you have overt mitochondrial free radicals and mitochondrial dysfunction. And then you're enrolled in the study, and you come into the CRU for 14 days, 15 nights. This is a pilot for 10 participants, 7 active, 3 placebos. It is randomized. It is placebo blinded. And then we're hoping to have an open extension that we're applying for right now for 6 months or to a year. And then this data, if it all goes as we have planned, then this helps us to provide merit to go into the Healy platform study in 2026 in 160 ALS patients. And then we plan on, we're focusing completely right now on ALS, but we do want to circle back to MS, Alzheimer's, and Huntington's. So the isoprostane data that it was a little bit of a gamble. We weren't sure it would work. We weren't sure if it would be a problem for us to do it. The reason why we take the urine is to hedge our bets that the cerebrospinal fluid will be even higher. We're going to want to screen people by isoprostanes through cerebrospinal fluid. So we use urine to hedge our bet, and it is working. So the first time we did it, we had 4 out of the 6 were basically in the 0.46, which is 99% confidence. One person had 2 dilute of urine, so they were out. They came back later on, and they were back in with a less, more concentrated urine. And then one patient participant is too low. Second run, we had ALS patients and MS patients, and secondary progressive MS, and we have people coming in for those. And yesterday we got this data that 5 out of the 7 patients are in for the next round. So they have already started to arrive. So this pharmacology, the pleiotropic effect, allows you to ask different questions than other platforms. So we know that MP101 DNP is going to completely flatten isoprostanes in these 2 weeks. If they flattens isoprostanes, and then antioxidants should be restored. We also know from work that we've done before that INOS, TNF alpha, and interleukins will come down in the spinal column based on our MS data. Others have shown that DNP induces neurite growth, and so there's a possibility of that pathway. And then Mark Mattson's lab at the NIH showed that mTORs reduce, autophagies changed, and our work and Mark Mattson's work showed that BDNF is induced. So these are the 8 different pathways that we're looking at in the cerebrospinal fluid in our patients in Europe. And then we'll look at a subset in blood to see how well some of them correlate to blood to CSF. So Mitochon was established literally 10 years, November 2014 from today. And so some of the patients, participants have ALS may wonder, where we've been. And the answer is that we've been trying to get to you. So in 2011, Robert Alonso, my co-founder, and I, we wrote our first ALS grant. And in 2012, we wrote 3 grants, and collectively till today, we wrote 14 grants to try to get a proof of concept study going for ALS and get into humans. But the elephant in the room is the fact that the drug was used 100 years ago at huge doses for obesity. And why would anybody want to give us money for diseases that are associated with wasting or weight loss when the drug was used for obesity? And the answer is that it has nothing to do with that, because 100 years ago, the drug was used at 300 milligrams per day, and we're using it now at 2 milligrams or 2.5 milligrams. So Paracelsus would say that all things are toxins, nothing not a toxin. It's dose that differentiates toxicity from remedy. And the fact is that MP201 or DNP has been put into mice for their entire lifespan at microdoses and improve extended longevity. So there's a complete difference between a high dose and low dose. But anyways, that's the elephant in the room that's delayed us getting funding. And so now we're on our way. So the hypothesis is
just 1 or 2 more minutes, okay?
Yeah, I'm done. So the basic hypothesis is that MP1's pleiotropic pharmacology and lowering ROSs, improving mitochondrial population, will reduce inflammation, ROSs, apoptosis, and reduce disease progression, while antioxidant status will be restored, BDNF will rise, cellular homeostasis will improve, neurite growth is possible with reestablishing lost communication, and the holy grail of improving mobility. So I just want to say that in consideration that no ALS patient or MS patient or ALS patients or animal models have ever been treated with a mitochondrial uncoupler today. And this is a completely novel approach. MP101 is the first small molecule to show functional recovery and paralysis. Its mitochondria are highly evolutionarily conserved. The mechanism of action is based on biophysics, so translation is superior to conventional drugs. And if we look at the timeline between how fast the mouse model advances and how fast human advances, I think that going to humans would be much better efficacy, because we have much greater time for attenuating progression. And of course, the point of interventions is key. So we're wondering if this could be a disease-modifying drug. So these are the papers that we've published since we established Mitochon 10 years ago. And The last slide is to thank all the people who have backed us, particularly Ben Franklin Technology Partners, who gave us the original seed money, and all the private investors, Cures Within Reach. And my hat's off to Dr. Lan Lepere for her coming to us and enabling this entire road going forward with her central hypothesis. Thank you very much. If there's time, I'll take questions.
Yeah, so we're just at time, but there are just a couple of questions. So a couple of quick ones I think we can knock out, and there are a couple in the chat that I might have you direct you to look at. But are you including all types of people with ALS, or just sporadic, or just certain types of familial ALS?
We haven't focused on familiar at all. Right now, we're focused totally on sporadic, the lion's share of patients.
Yes.
So that's where we're at right now.
And I guess to follow that up, would you plan to test people and exclude them if they had an ALS causative mutation, or just sort of be mum on it, and anybody with ALS could enroll, which would mean the majority would be sporadic?
Right. I think in these pilot studies we're doing now, we would keep it sporadic, but as it opens up to the Healy platform trial, there's no reason to not. We would do want to get people probably earlier onset while they're still ambulatory. We want to make sure they can take the capsule, these things. And we would also include isoprostane screenings.
Okay. Then there was a question about, after people finish phase 1, will there be an open label extension of any kind? Can people stay on the drug?
Yeah, that's exactly what we're looking for right now. So the study that's going on in Europe, we're asking for that open extension for a year right now. And then the Healy platform study would be 1-year open extension as typical.
And any side effects of MP101 on cardiac mitochondria that you know of?
So MP101 at these doses has been shown to be protective. We've done many models, 10, 12 different models, and it collectively is pan-neuroprotective. So if anything, the cardiovascular risk would be improved. And it's been put into various models and shown to be protective.
Terrific. So one last one. Decreasing mitochondrial calcium at the cost of increasing cytosolic calcium is expected to be toxic based on the calcium field. So what are your thoughts about this?
Yeah, so we've discussed this before because we have other data, and we work on TBI and with MP201. So the basic idea, I think, is that calcium, when it rises in the cytosol, will likely leak out and go someplace else. But I would have to say that in the longevity studies we have done, which and others have done, there's actually no issues in terms of these microdoses on longevity or cardiovascular risk. I think that the studies are out there that suggest that it's lowers free radicals in the heart, in the muscles, in the brain, and is all moving towards pro-survival.
Very good. Well, thank you so much, and thank you for answering the questions. Got to most of them. There may be a couple more in the chat as well.
Thank you, James.